Um, I'll just get started. If anyone else wants to join, I'll let them in later. My name is Nandika. I'm a PhD student at ISC Bangalore, and I will be discussing some content related to week 8 of experimental biotechnology, where in this week we have discussed a new topic, which is cell biology, how to maintain cells, and a small part of differential centrifugation. So I'll just be discussing some topics and some problems related to that. I hope you can see my screen. So, as I mentioned earlier, in this week we've discussed topics of cell biology. So, we'll first see what are the basics of cells. So, as we all know, cell is the basic unit of life, and the or first cells on Earth were these prokaryotic or cells that we see today as bacterial cells, where prokaryotic is basically pro meaning before and karyon meaning nucleus. So it is basically s- cells without a nucleus, right? So you will see instead this region called the nucleoid, where the genetic material is floating around here without any defined organelle to contain it. Um, they also have, obviously, the cell is made up of this matrix called the cytoplasm, in which all the other ribosomes, proteins, the nucleoid, etc., are suspended. And they may have different inclusion bodies or other such small regions within the cytoplasm. Of course, the cell boundary is defined by a membrane. So you'll have a lipid bilayer, which is defining the cell wall, sorry, cell membrane. And outside of the cell membrane, there may or may not be a cell wall. Okay. And again, prokaryotes, as means of locomotion, may require some sort of membrane extensions from the cell wall. So this will be things like flagella, pili, fimbriae. So these are all extensions of the cell membrane, which we can see in prokaryotic cells. These are some common shapes that we can see in prokaryotic cells. They can be round, in which case they're called cocci. They can be rod shaped, so it's slightly elongated. So these are usually called bacillus. If this rod has a slight curve in it, okay, so it's kind of like a comma, right? Comma shaped cell, it's called vibrio. You may be familiar with Vibrio cholerae, it's similar. A short rod, right? So it's kind of halfway between a round sphere and a rod, is Cocobacillus, right? Makes sense. It has features of both of these. If it's spiral, it's called spirillum. And if it's long, loose helical spiral, okay, so I think you can see quite easily a difference between these two. This is kind of a loose spiral, right? This is a loose spiral, and this is a more contained spiral. So this is a very long spiral, is a spirochete. Okay. And again, they can be given different names depending on how they form or how they associate with each other. So if your bacterium is existing as just a single coccus, it is a coccus. But if two of them, if they exist in these pairs, they'll be called diplococcus. Four of them together will be called a tetrad. Okay, just these four arranged in a square. But if this diplococcus chain is elongated, and now I get a chain of many cocci, that is something called streptococcus. Instead of growing in a chain, if instead they grow as a cluster, then it is called staphylococcus. Right? You may be familiar with some of these species also. Things like Streptococcus pneumoniae or Staphylococcus aureus. So that is just referring to the way in which the cells are growing. Similarly, if Remember, bacillus was what was referring to a rod. If we have a chain of these rods, it becomes a streptobacillus species. Okay, yeah, I won't talk about this. Um, so, in contrast to the prokaryotic cell, we also have a eukaryotic cell, right? So, U meaning true and karyon meaning nucleus. So, these are cells which have a nucleus, which is this organelle here, which is a membrane bound organelle containing the genetic material. You can see this consists of this membrane, the nuclear envelope. So this is double bilayer. The nucleolus, and within the nucleolus, we have the chromatin and the genetic material of the cell. And things can move inside and out of the nucleus through these nuclear pores. Like you can see, the boundary of the eukaryotic cell is also defined by a cell membrane. So this is again a lipid bilayer. Some eukaryotic cells, such as plant cells, will also have a cell wall surrounding the cell membrane to give additional structure. But what you will see additionally in eukaryotic cells are all these other subcellular organelles which are absent in prokaryotic cells. So these are all membrane bound subcellular organelles, such as mitochondria, the Golgi body, chloroplasts in plant cells, and so on. And those are another defining characteristic of eukaryotic cells. Just as to summarize what the difference between these two are, you can see that the eukaryotic cell is much bigger than a prokaryotic cell. Okay. Because of this, the prokaryotic cell, since the cell is smaller, the surface area is larger for the same volume. Right? Whereas the eukaryotic cell is bigger, so the volume is large compared to the surface area. Obviously, by definition, the prokaryotic cell has no nucleus. The eukaryotic cell has a nucleus. Um, the genome of a prokaryotic cell will contain one single chromosome without histones. Archaeal cells may contain histones. This will be a single circular chromosome. Whereas, as we're familiar with eukaryotic cells such as our own cells, they will have multiple chromosomes with histone proteins. Cell division in case of prokaryotes will take place by binary fission. The cell is surrounded by a membrane made up of lipids, but the lipid composition will change depending on the type of cell. So, archaeal cell walls, especially, have a lot of uh, adaptations to grow in extreme environments, which is why you'll see there are many differences between archaeal cell walls and bacterial cell walls. Okay, these are all adaptations that are made to grow in places like extreme heat or extreme pressure. Eukaryotic cell walls will characteristically have sterols such as cholesterol, which will be absent in bacteria. Cell wall composition, as we discussed, the bacterial cell wall, either they will have a peptidoglycan cell wall or they will have no cell wall. So that is basically defining gram positive and gram negative bacteria. The archaeal cell wall can have many different components. Like we discussed in eukaryotes, animal cells will not have a cell wall, but other cells such as plants and crustaceans, some algae, and so on will have a cell wall. The most common in plants will be a cellulose cell wall, which is basically giving the cell more rigidity. Right? Cellulose in plants, chitin in insects, so on, which is giving the cell more rigidity and mechanical strength. Motility structures. These are the membrane extensions that we discussed. So that will be things like the flagella in bacteria. Are they membrane bound organelles? Yes, in eukaryotic cells. No, in prokaryotic cells, obviously. It's an endomembrane system. So this is referring to membrane extensions within the cell. So that will be things like the endoplasmic reticulum and Golgi bodies within eukaryotics. There is no such system within prokaryotes. Even the composition of the ribosomes is different in these two classes. Prokaryotes have 70S ribosomes, while eukaryotes have 80S ribosomes. 70S in mitochondria and chloroplasts. And the theory behind the fact that mitochondria and chloroplasts are more prokaryote like is something called the endosymbiont theory, where it basically claims that mitochondria and chloroplasts come from ancient bacteria, ancient cyanobacteria, right? So it's an ancient cyanobacterium that got incorporated inside a mammalian cell. And over time, it has lost some of its features and has become an essential part of a mammalian cell. But since it came from a cyanobacterial origin, it retains some bacterial features, such as the nature of the ribosomes. Okay. So now that we have a basic idea of what cells are like, obviously these cells, such as if we start discussing microbial cells, they're growing quite happily in the environment, but maybe we want to grow them for our own purposes in the lab. So for that, we need to know how to grow these cells properly and how to properly support their growth. So here you can see the nutrients that are required for microorganisms. You see that there's a selection of some nutrients that are essential for all microorganisms. That is basically hydrogen, carbon, nitrogen, oxygen, phosphorus, sulfur, and selenium. Okay, as you can see here. Selenium is required in a very trace amount, but carbon, nitrogen, oxygen, phosphorus, and hydrogen are required in quite appreciable amounts, which means that if we want to grow cells separately outside of their natural environment, we have to provide sources of all these elements. We have to provide some carbon source, we have to provide some nitrogen source. Oxygen and hydrogen also need to be present, so there needs to be some way for air to come in contact with our culture, right? Phosphorus, depending on the kind of culture we should also have some source of phosphorus and sulfur. Okay, you'll see there are also some essential cations and anions for most microorganisms. Things like sodium, magnesium, potassium, calcium, chloride. These are common ions that we have to keep to maintain the osmotic balance, maybe because they're required in some enzymes, so on, but they are required for most microorganisms. We also have some trace metals, and some of which are essential for some microorganisms. So these are such as these transition metals. These are often used as cofactors in enzymes, and hence they are required by microorganisms. All sorts of some other very trace elements that can help in certain circumstances. And so we have to keep that in mind when we're creating a medium to support our microorganism growth. Yeah. So here we see micronutrients needed by microorganisms. So we already discussed what are the macronutrients required, which were calcium, hydrogen, oxygen, nitrogen, phosphorus, sulfur. But there are also some micronutrients which are required, which are listed here, like boron, chromium, cobalt, copper. Right? You're seeing copper is required in respiration, cytochrome C oxidases in photosynthesis, and so on, because it is a cofactor in these enzymes. Iron, again, it's a cofactor in these enzymes. Manganese, molybdenum, nickel, selenium, etc., zinc. Right? I'm saying not every micronutrient is required by all cells, but they are required by the majority of cells. So, if we want to create a culture medium, we should have a balance of all these elements within it in a suitable ratio so that it can support their growth. So, if I want to have defined culture mediums, where I am defining the concentration of each component in the medium. For growing a simple organism like E. coli, then I can use a mixture of several salts. So you can see here these are going to provide the ionic components of our solution, and they're also going to maintain the osmolarity of the solution, right? It should not be that the ionic strength of our solution of our medium is very different from the ionic strength, very different from the ionic strength of our cell. Because in that case, we will get a phenomenon called osmosis, which will cause sorry. So if the ionic strength of our medium is very different from the ionic strength of our cell, then what will happen is that we will get movement of solvent either inside or outside the cell. That means either water will be dragged out of the cell or water will go inside the cell, right? And that will either cause the cell to swell or will cause the cell to become extremely shriveled and shrunken. So that is not going to help support bacterial growth, as you can see. It'll cause cell death. So it is very essential that we maintain the osmotic balance of the medium. Okay? So that's what all these salts are going to help doing. You can see that here we're going to provide potassium, magnesium, calcium, right? We're supplying phosphorus, we're supplying sulfur, supplying some ammonium. So this is going to be a nitrogen source. Glucose. So this is going to be a calcium source. Okay, essentially what is going to provide energy for bacteria. Trace elements, as we discussed, all these metal ions, distilled water, pH 7. Obviously, if you want to maintain the pH of the culture medium, we should have some buffer. So here you can see we have introduced a phosphate buffer. Yeah. So this is a very simple basic medium that we can use to culture a simple organism like E. coli. Now, E. coli is easy to grow because it doesn't have many like stringent nutrient requirements. If instead we wanted to grow some very fastidious organism that required very high nutritional requirements, like say this leuconostoc, okay, this is a different bacterial species, where it has very strict nutritional requirements. You can see that suddenly the medium has become a lot more complex, right? In addition to all these salts, now we have to add amino acids, purines, pyrimidines, vitamins, and so on. Right? So what we are doing is essentially we had to make this medium, right? You can see that this medium would be extremely difficult to make because you have to weigh all of these components individually and add them in. So rather than going for defined culture media, so usually we will go for something called a complex culture medium. We'll be keeping these things such as yeast extract and peptone. Okay? Yeast extract and peptone are not chemically defined components, right? They are components coming from an organic source which will contain all of these. Essentially, we are replacing this defined composition with these two components from an organic source which will contain all of these components but maybe not in this defined amount. Okay, because we don't know the exact composition of yeast extract and peptone, but empirically we know that at this concentration it is sufficient to provide nutrition for our bacteria. Okay, so in this complex culture medium we have our carbon source. This is going to provide the nitrogen source as well as any other trace elements. Okay, this is potassium, distilled water. Right, you can see this is now a much easier medium to make compared to this. Right? And of course, if there is some special requirement for any nutrient, you have to add that in separately. So this is something called Thiobacillus thioparus. From the name itself, you can understand it's a sulfur metabolizing bacterium, right? Thiobacillus. So we have to add some additional sulfur as part of this media composition. Yeah. And here you can see another special thing is that it does not need any alternate carbon source. It can take CO2 from the air. Okay. And just visually, this is a defined medium where we've just added powders of all of these components. And this is a picture of the complex medium where we have added these components whose composition is not defined. And just visually, you can see that there's a big difference between the two. Yeah. Complex medium is acquired color due to various organic extracts and digests. Now, bacterial culture media can be classified into various forms. In its physical nature, here we saw how to make a liquid medium. Right? So here the bacterial cells will be floating in this medium and they'll grow in this sort of suspension. But of course, we can also solidify the media and make it a plate. Right? So within the plate, and then we have bacteria growing on the surface of the plate. So that we can do by adding something, a gelling agent called agar. And when the agar solidifies, it causes the media to take on a solid form and we can grow bacteria on the surface. A chemical composition, as we just discussed, we can have simple or basal media, which is just the basic requirements, okay, nothing fancy, just the basic requirements for growth. Defined or synthetic media, like we discussed, this is a defined medium, that where we know the exact amount of each component. And complex media, where we have used other organic components where we don't know the exact composition. Another classification of culture media is based on their purpose. So obviously we can grow media, just have media just for growing bacteria, but we can also have enriched media where we will give a larger amount of some nutrient because we want we want some effect for it to show some effect in the culture. Selective media, which will allow some kinds of bacteria to grow but other kinds of bacteria will die. Differential media, where different kinds of organisms will show some physical difference when they're growing in their media. Indicator media, transport media, anaerobic media. So these are all different kinds of media depending on what kind of bacteria we want to culture. Right? So just basic liquid media that we often use in the lab will look something like this. So this is glycerol, yeast extract, tryptone. This is just extremely basic medium. Glycerol is the carbon source. Yeast extract and tryptone are providing all the other nutrients. And here they're saying all you have to do is weigh this, dissolve it in water, and obviously we only want bacteria of our interest to grow in our media, so we need to sterilize it. So sterilize the medium by passing it through a pre-rinsed 0.22 micron filter. So 0.22 micron is smaller, is basically the pore size of the filter, and that is smaller than all bacteria. So no prokaryotic cells will be able to pass through a 0.22 micron filter. Yeah? So this will make sure that the medium is sterilized. This is probably the most common medium that we use in labs around the world. It's called LB, or Luria bertani. So it's commonly used to grow organisms like E. coli and so on. So LB medium actually has three components: tryptone, yeast extract, and NaCl. NaCl is here to obviously provide ions as well as maintain the osmolarity of the medium, like we discussed. The medium should not be hypertonic or hypotonic for our cells because that will lead to cell death. Tryptone and yeast extract are nutrient sources. And here it says first you have to dissolve it in water, adjust the pH to 7, adjust the volume, and sterilize by autoclaving. Yeah. So the autoclaving will heat it for 20 minutes at 15 psi, 121 degrees Celsius, which will kill any growth that could occur. And then we have prepared sterile media that we can use for culturing basic microorganisms. Now, as you can see, this was a complex medium, right? LB is a complex medium because tryptone and yeast extract are not defined components. Instead, we can, if you want to have a defined medium, then one example of that is called M9 minimal medium. Okay, this is basically just the minimal requirements for growth, bacterial growth, where we have different salt solutions. This is sodium phosphate, potassium phosphate, NaCl, ammonium chloride. Magnesium sulfate, carbon source such as glucose, calcium chloride, and water. Okay. So you can see this is basically a solution of different salts and a carbon source, sterilized by autoclaving, and so on. So this is just the basic minimal medium, and obviously if you want to supplement anything into this, then that will be over and above the minimal medium. Now, if you want to grow a lot of bacteria, okay, you want the bacteria to be really healthy, produce a lot of protein and so on, you can use something called terrific broth, also known as TB, which you'll see it has the same components as luria broth, okay, it just has this higher amount. Okay, so TB sometimes is also, so you can also sometimes use 2x LB, which is basically double the LB recipe. Yeah. So this is essentially an extremely nutritious medium that supports very high bacterial growth and protein expression. Okay. Um, Again, this same deal, you have to make it up in water, sterilized by autoclaving. And like we discussed, we can obviously make media for bacterial growth, but we can also make different kinds of media. So when we start adding other components into the media, we can create media that have different purposes. So this is something called selective agar medium. And what selective means is that it only allows some types of microorganisms to grow and prevents the growth of other microorganisms. So this is selective for Campylobacter. Okay, so all these streak bacteria are Campylobacter. Types of bacteria are not able to grow on this. Most common also have things like called differential media, which means that different bacteria grow in different ways on the media. This is something called McConkey agar. Okay, on this plate you can see we have streaked two different kinds of bacteria, and one side of the plate is turned pink, and the other side of the plate is yellow. And this is because if we look at the composition of McConkey agar, we see this extra carbon source lactose. Okay, so what actually happens is that if a lactose fermenting bacteria grows on this kind of plate, then by product of a lactose fermentation, you'll see this indicator neutral red causes this indicator to change from yellow to pink. Okay, this is a lactose fermenting bacterium and this is not. There's also, so this is why it's called a differential medium, because these two bacteria are showing different phenotypes, not different phenotypes, but they have different effects when grown in this media. But here you also see it's called a selective medium. It's called selective because it is actually selective against gram positive bacteria. So it contains something called bile salts, which do not allow gram positive bacteria to grow in the medium. Okay, so it's also called selective medium. One of the most famous differential media, something called EMB agar, is eosin methylene blue. And what happens is when you streak something like Again, this is selective for gram-negative bacteria. And when you grow something like E. coli on EMB agar, you will see that actually it starts turning green. Okay, so this sort of green growth on EMB agar is selective for E. coli. So, so when we start going into things like diagnostics and uh, searching for pathogens and so on, this is a very useful way in which media can be modified to, to suit the purpose of our experiment. So obviously, as we discussed, bacterial cells divide by binary fission, which means that each cell divides into two new cells. You have two daughter cells, right? And so in each generation, the cell number is increasing by the power of 2. Right? So in generation 0, we have 2 raised to n cells. So here, generation 0, 2 raised to 0, we have one cell. The one cell has split into two, so now we have 2 raised to 1, 2 cells. Here we have 2 raised to 2 or 4 cells. This is 2 raised to 3, which means 8 cells. Okay? So with bacteria, it is quite easy to estimate, I would say, the number of cells which are there in the bacterial population. Right? It's possible to predict the number of cells in a population when they divide by binary fission at a constant rate. Right? Because the generation time or the time required for one cell division for most bacteria is known. Let us consider single cell which divides every 30 minutes. Right? Which means generation time is 30 minutes. Number of cells is increasing exponentially. Can be expressed as 2 raised to n, where n is the number of generations. Right? If cells divide every 30 minutes after 24 hours, 48 divisions will take place. Right? Because in every hour we're getting 2 divisions. Applying this formula, after 24 hours, we'll have 2 raised to 48 cells. Okay. Which is basically 2.8 into 10 raised to 24. 2.8 10 raised to 14 cells. Okay. Obviously, you're probably not going to start bacterial culture from one cell. There'll be some initial number of cells. And so the formula can thus be that number of cells at time n is n0, which is our initial number of cells, multiplied by 2 raised to n, right? Where n is number of generations. Now, this division process will go on indefinitely as long as there's nothing to stop it. Now, when we're growing these cells in culture, in a flask or something, then there's only a limited amount of media which is present in the flask, right? We're not adding more and more media as the cells are keep growing. So that means even the number of nutrients that is present in the flask is limited. So at some point, that will start giving pressure to the growing cells, and as they start consuming more and more of the nutrients, there will not be that much nutrition left for the new cells. So when we grow bacteria in culture, the number of cells will actually follow a graph that looks something like this. Okay. First, we have something called the lag phase. Okay, so this is when the bacteria are sort of acclimating to the medium. Okay, they're preparing for division, so on. Once the bacteria are ready to divide, this is the lag phase. The first portion is called the lag phase, because in this phase there's no increase in the number of living bacterial cells. Okay. Once the cell number starts increasing, we will enter something called the log phase, which is here, where the cell number is increasing rapidly over time. So this is the region in which the cells are dividing regularly every 30 minutes. And so every 30 minutes we will get an increase of basically it'll increase by a factor of 2. Okay, and this will go on until the nutrients within the medium will start getting exhausted. So once the nutrients are exhausted, that will cause the cells to come out of the log phase, and we will enter something called the stationary phase. Okay, within this phase there is not much change in cell number. It means the rate of cell division and cell death are roughly equal. Okay, and over time, after all the nutrients are exhausted, what will happen is that the number of cells will start falling, right? Because cell death is taking place, but cell division is not taking place at the same rate. So the culture will enter the death or decline phase, where we have exponential decrease in the number of bacteria. Obviously, if we want to do anything with the bacteria, say we want the bacteria to express our protein or something like that, then we would want it to take place in the log phase. Right? Because this is where the bacteria are healthy, they are dividing, they're growing well, they're not facing any nutrient scarcity or anything. So that's why normally if you are going to induce the culture to produce a protein, right, you will normally do it somewhere here in the log phase. You might have heard that you grow your culture to a optical density of zero 0.8 or 0.6, and then you ask it to produce, then you induce it. That's because the culture has reached the log phase, and that's a good time for the cells to start producing protein. So how we measure the nature of what stage the culture is in is we can use something like this, which is spectrophotometer, where we will just measure the turbidity of the bacterial cell suspension. Right? What is turbidity? Turbidity is basically just scattering of light from it. So here you can see this is just culture medium. Okay, there are no cells in this. So what is going to happen is that yeah, I just have a tube. It has liquid. Okay, and the liquid, there's no actual particles within this liquid, so light is able to pass through it and it'll be detected over here. Instead, if my liquid is full of small particles, okay, we'll call it cells, and I'm giving light of appropriate wavelength, okay, wavelength of the light which is similar to the size of the cells, what is going to happen is that this light will interfere with the cell, right, where it's incident on a cell, it will get scattered. Right? And as a result, the amount of light which is able to travel straight through the culture is less. Right? Because a lot of it has been scattered while it was traveling through the culture. So that is going to result in less light which is reaching here. So this difference between these two measurements is going to give you something called the optical density of the sample. Optical density or turbidity. Normally for bacteria, like you can see, the wavelength of the light that we use should be comparable to the size of the particle that we want to see. So normally for bacteria, we will check OD at 600 nanometers. Okay. So where you can pick up your culture, uh, take a small aliquot of the culture, take it to spectrophotometer, check the OD. If the OD still is like 0.1 or 0.2, that means the culture is still in the stationary phase. Maybe it's starting to enter the log phase. Okay, so at this time, we don't want to disturb the bacterial culture. But once it reaches an OD of something like 0.6 or 0.8, that means the cells have actively reached the log phase. So we can start using the culture for purposes like protein expression, competent cell preparation, things like that. Mm, obviously, we discussed. So here we have discussed just how to grow normal bacteria like E. coli, okay, which have very regular culture requirements. But of course, there are different kinds of bacteria. So there's some bacteria which require a lot of oxygen, some bacteria which require complete absence of oxygen. Okay, these are anaerobic bacteria. Some bacteria can survive at various concentrations. These are facultative anaerobes, where if the possibility of anaerobic growth is there, they will take it. Air tolerant anaerobes, which can grow with some exposure to air. Microaerophiles, which have requirement of very less oxygen. Okay, so obviously these are all factors you have to consider when you think about how you want to grow your bacteria. So if you're growing something which is a more anaerobic bacteria, maybe you need to flush your incubator with nitrogen or something like that. These are considerations you have to make. Then, of course, what temperature should you grow your bacterium in? Again, E. coli is a human pathogen, so it grows best at 37 degrees. So it is something that would be called a mesophile. Okay, these are bacteria which grow well at a kind of mid temperature range, which is kind of room temperature, human body temperature, and so on. It's like 20, 30, 40 degrees Celsius. But some bacteria like to grow at higher temperatures. So it all depends on what the natural environment is. So, for example, there are some bacteria like Pyrococcus, like which naturally grow in volcanic vents and things like that. And you can imagine those are very hot places. So they grow best at extremely high temperatures, like 90 to 100 degrees Celsius. Also, there are some bacteria which grow well at cold temperatures. They're called psychrophiles. Okay, these are growing well at around 10 degrees. So, yeah. So, all these bacteria have different adaptations within their own cells that help them to survive at these different temperatures. And it's important to know what is the correct temperature to grow your bacterium of interest. They also have different pH requirements. So, again, like E. coli grows in human body, so it requires this environment like human body, right? So, it's a neutrophile. It's happy around pH 7. But of course, you get bacteria which are happy at other pHs. Right? So that those who like to grow at low pH are acidophiles. Those who like to grow at higher pH are alkaliphiles. Okay? Acidophiles have optimal pH around 3. Alkaliphiles have optimal pH above 9. Okay, we can discuss some questions related to this part. So, here says the organelle common to both prokaryotic and eukaryotic cells is dash. So, mitochondria and chloroplasts and Golgi bodies. Yeah, Aditi, go ahead. Yeah, you can feel free to either type it there or unmute yourself and answer the question. That's fine. But Aditi is right. The organelle common to both prokaryotic and eukaryotic cells is ribosomes. All of these are present only in eukaryotic cells. Okay. The medium component responsible for osmotic balance in LB broth is dash. So, um, remember the medium component which is responsible for osmotic balance specifically is NaCl. Okay, these tryptone and yeast extract and trace elements are responsible for providing sorts of undefined nutrition and so on. But primarily, NaCl is responsible for osmotic balance. Okay. M63 and M9 media are used respectively for dash. So, these are minimal culture media that are used in microbiology. They are not used for cultivation of mammalian cells, not used for growth of plasmids. They are usually used for the cultivation of E. coli strains. During bacterial media preparation, when should antibiotics be ideally added? Okay. So so we add antibiotics sometimes to bacterial media when we want to grow organisms of our interest specifically. So I want to grow some bacteria in which I have put a plasmid of my interest, and I know that my plasmid has ampicillin resistance. So that means my bacterium can grow in an ampicillin containing medium, and the other surrounding bacterium cannot. Right? So that is going to select specifically my bacterium. The problem is most antibiotics are heat sensitive. So when we make the media, obviously to sterilize it, we're going to autoclave it. So it's going to be heated to 121 degrees Celsius. When we take it out of the autoclave, it is going to be extremely hot. And if we add the antibiotics at that point itself, the antibiotics will be degraded. So they will not be able to have any effect. Right? So there's no point in adding them when the media is hot. Also, there is no point adding them just before autoclaving the media because then they will be degraded in the autoclave cycle itself. Okay? So ideally, we should be adding it when the temperature is less than 50 degrees Celsius. Okay, bacterial cell wall is composed of dash. So, as we discussed, cellulose, chitin are components of eukaryotic cell walls. The bacterial cell wall is composed mainly of peptidoglycan. Okay, yeah. So this was the section on how to culture bacterial cells, right? But obviously, um, we can't. If we want to study phenomena that are taking place in eukaryotic cells, then we would want to culture those as well. Okay? And eukaryotic cells present a very different sort of challenge for culturing because culturing bacterial cells, so like the standard bacterial strains are quite easy to culture actually because you just have to inoculate them. Once they reach a certain cell density, you add them to a bigger flask, and so on. So it's not particularly difficult to culture bacterial cells, but eukaryotic cells can be challenging. Okay, for a number of reasons. And the first reason is if you consider cells in the human body. Okay, most cells. Okay, we won't talk about things like blood and so on, but most cells they stay in one place, right? They are sort of adhered to some to a basement matrix or basement membrane, right? So they form connections with the basal layer, and they can also form connections with each other. Okay, so this is an example of epithelial cells, but of course there are other cells in the body like this, where they have these things called cell adhesion molecules. They have things called cadherins. They have integrins, and they have proteoglycans, and these are all responsible for anchoring the cell in one place. So you can see that naturally the cell requires some sort of support for it to grow. So when we grow these cells in our lab, we also have to provide some support for it to adhere. It has to stay stuck to the surface so that it can grow in its natural morphology. Okay? So when we grow cells in a lab, we have to use some sort of dish, either like this. Okay, so these are all wells, and we will grow cells on the bottom surface of the well over here. Okay, the cells will be stuck here. So you can have wells of different sizes. So a 6 well plate, and this is a 24 well plate, this is a 96 well plate. We can use a petri dish where this whole bottom region will be coated by the cells, or you can use a flask. So the flask is giving you a big surface area where the cells can attach. This cap will close tightly, and might be a filter in the cap which will allow for air to exchange properly. So Normally, if we want to grow cells in plastic like this, then we have to treat the plastic surface so that it is suitable for the cells to attach to it. So when you buy these materials, they will say tissue culture treated. So that means that they've undergone plasma treatment so that these surfaces have actually become more hydrophilic. Okay? They're less hydrophobic, they are more hydrophilic. They've imparted some charge on the surface which is going to help the cells to attach properly. So yeah, as we discussed, cells in a flask will look like this. Cells are stuck here. This is cell culture medium. Yeah, this is the cap of the flask. But there are also some cells which have adapted to growing in a suspension culture. Right? So this is how we would grow bacterial cells. We would have some liquid culture. The cells are growing in the suspension. We would just keep it in an incubator like this. Right? So this is called a suspension culture. So this requires some specialized cell lines, such as this, what is called HEK293 FS. This is a cell line which is specialized for growing in a suspension culture. So, as you can see, there are two main types of mammalian cell culture: adherent and suspension. Adherent is appropriate for most cell types, including primary culture. Primary culture is when you directly take cells from an animal or from the tissue itself. You isolate the cells from the tissue and you try to grow them. But the suspension cell culture requires cells which are adapted for that suspension cell culture, which means that we have gotten rid of its requirement to adhere to a surface. Or cells of hematopoietic origin, right? So cells such as blood cells which are naturally growing in suspension manner, those we can grow in a suspension cell culture. Um, Obviously, since we are growing it on the surface area, the growth is limited by the surface area of the flask. The flask should be cell culture compatible. No agitation is required because it's just growing on the surface. Okay? But because it is growing on the surface, it can only grow as long as there is surface area available for it to grow. Okay? So if my flask is like this, so I'll start with some small number of cells. But as they keep growing, okay, they will start filling up the surface area. Okay? And once this surface area is covered, okay, they've covered the full surface area, now there is no more place for them to attach, right? And because these are adherent cells, if they cannot attach to this culture, this is the dish surface, they cannot survive. So it is necessary, once we have reached this number of cells, it is necessary for us to remove the cells, okay, to remove the cells from this surface and add them to a new surface. Okay? So we'll take all of these cells, we can split it into multiple cultures and add them to new flasks. So all the cells now get proper space and nutrition, okay, and all they will be able to continue multiplying. So this process is called passaging. Okay. Passaging is basically this process of removing the cells from one culture vessel and transferring them to another. And for that, we have to get them to detach from the surface. And that can be done either by an enzyme, such as trypsin, or mechanically, which means we can scrape it away from the surface. Okay? Trypsin is basically going to digest all the adhesion molecules that were produced by the cells that have adhered to the surface. So this is all about adherent cell culture. If we talk about suspension cell culture, uh, it is easier to passage, so you don't have to detach it from the surface to add it. You can just This is growing in my flask. I have this many cells. Cells are growing like this. At some point, the cell density will become too much, just like a bacterial culture. It'll start using up the nutrients, and there won't be enough nutrition left for cells to grow properly. So, what we can do is add more media. Okay, we can just add more media to this, and so that will allow the cells to start dividing again. Okay, so you can see it's much easier to passage. Okay, so it does not require enzymatic or mechanical dissociation because it is present in the suspension itself. Growth is limited by cell density, right? If the cell density becomes too much, then cells will start dying. Can be maintained in any sterile culture vessel because it does not require the cells to attach properly. Requires agitation. So this is just like a bacterial culture. We need it to be exposed to the air, and it requires agitation for gas exchange. So when you would want to use a suspension cell culture is for something like large scale protein production. We want to do something as a big batch. You can imagine how many cells will be present. Okay. Say I have a 2 liter flask. Okay. Flask is 2 liters. I have 800 ml culture in it. Right? I have 800 ml culture. And the cell count of this culture will be 2 into 10 raised to 6 cells per ml. Right? So the final number of cells in this flask, okay, this single flask, will be 1.6. Right? I'm getting 10 raised to 9 cells from a single flask. So you can see this is very good for things like bulk protein production or anything that needs to be done on a large scale. Okay, it's easier to maintain this. Whereas this is better for, obviously, if you want to do things like microscopy or things on a small scale, then adherent cell culture is appropriate. So, as eukaryotic cell growth curve, you can see it's very similar to the bacterial growth curve that we just saw. There is a stationary or a lag phase in the beginning. Once the cell is in a position to start dividing properly, you will get the exponential or log phase. And at this time, we have to feed the cells, right? Which means we have to add media, change the media, and so on. And once it reaches the limitation either of space, media, or something, it will enter the plateau phase. And before it enters the plateau phase, it's the good time to passage it or subculture the cells. Okay. So, like we discussed, this is what we call subculture. Subculture from monolayer. So, monolayer means it's growing in a single layer on the surface of the flask. Okay. So, here you can see the monolayer has completely covered the surface. Okay, there's no more space for it to grow. This is the media on top of the monolayer. So, how we want to subculture this is first we'll remove the media and wash it once. Then we'll add trypsin. What trypsin does is that it digests all the cell adhesion molecules that are present on the surface of the cell. So this is going to destroy any cell-cell adhesion. It's also going to destroy the cell adhesion with the surface. So after we incubate it with trypsin, what is going to happen is cells are going to detach and you will get these round cells. Okay, these are round because now they have not taken any particular morphology. So the round cell means it's not attached anymore. So what we can do is resuspend these cells in the medium and just seed them in a fresh flask. So here you can see we have these round cells in the fresh flask. After they adhere properly, they will take their proper morphology. But now because the cell number is less, they will have proper space to grow. So this is a continuous process. once these grow, again we will come back to this first stage, okay, where we have to get this monolayer of cells, where again we have to subculture it. Okay, so in cell culture, this is a continuous process. You have to keep maintaining your cells so that they stay healthy and they don't die. Okay, so this can be a very laborious job. But that you cannot subculture the cells indefinitely. Okay, so mammalian cells, most mammalian cells will have something called a Hayflick limit, which is a limit to the number of times you can subculture it. Okay, so here you can see all the different the subcultures, and what actually happens is after a certain number of cell divisions, most cells will start dying. Okay, so what will happen is that most cells will start going into senescence and cell death. Okay, because over this process they will essentially hit the limit of how many times a cell can divide, and that will uh, mean basically you cannot maintain the cell line in culture anymore. Okay, it will start dying. There are some special cell lines that are called continuous cell lines, and what that means is that they have surpassed this limit, and when they've surpassed the limit you can continue maintaining them indefinitely. So most cancer cell lines will be continuous cell lines. Okay, and there are also ways in which you can make other regular cells into continuous cell lines. Okay, it's called an immortalization process. But basically, most primary cell lines will have a limit on how many times you can subculture them. After this limit, what you have to do is go back to the beginning here. So you have to revive a new set of cells which are at this stage, and then again you can culture them until they hit this limit. But if you have a continuous cell line, then you can keep maintaining it for as long as possible. Yeah. Um, One thing that can differentiate a finite from a continuous cell line is just a continuous cell line will obviously have many differences, right? Because it is basically now an immortal cell line. So one of the differences is in chromosome number. This is a continuous cell line. It's a metastatic melanoma. You can see it's a cancer cell line. And instead of having the normal 46 chromosomes, you can see it has a highly increased chromosome number. It's one of the factors that is contributing basically to its continuous nature. Okay, so now we saw how we can maintain cells. We can discuss now what are the components of cell culture media. So the idea is basically the same as bacterial cells. You need one carbon source, right, which is source of energy. You should have, right, because our cells need proteins and so on. We should have amino acids and vitamins. We should have balanced salt solution. Like we saw, we need ions like sodium, magnesium, potassium, and so on. So it is necessary to have these ions to maintain optimum osmotic pressure. Also, we need essential metal ions to act as cofactors and so on. Phenol red dye is something that is used as a pH indicator. We'll discuss it a little bit later. And we also need a buffer, right? So it should stay in the pH range which we want. Typical growth conditions, if we are growing human cells, then it should grow best at human body temperature 37 degrees Celsius. And most cell lines will require us to maintain 5% CO2, okay, for their growth. And we'll discuss what role the CO2 is playing. So, just talking about the pH, most cell lines grow well at pH 7.4. They're quite tolerant to a little change in pH. So, it might be that your cell line might grow better at like pH 7.7, but it will still grow at 7.4. So, most media that you get on the market will be formulated for this pH. And how you can tell the approximate pH of your media is by using this indicator called phenol red. Okay, this is a pH indicator and it can take on a number of different colors depending on the pH of the media. So if your media is in its ideal form at pH 7.4, you'll see it as a red color. But as the media starts becoming acidic, it will start becoming orange and then eventually yellow. On the other hand, as the media starts to become basic, it'll start becoming pink. Okay? So it'll give you like a visual idea of what the approximate pH of your cell of your media is. And if your media is looking like this or like this, then you should not be using it for your cells. Okay, it's not going to support your cell growth. Also, if your media starts turning acidic, it can indicate that you have some contamination in your culture. Okay? So phenol red is very useful just as a visual indicator of if your cell culture media is good to use or not. Another important thing you should know about mammalian cell culture media is that the buffer is maintained in most media is maintained by carbon dioxide. So carbon dioxide is used to form to as a part of the bicarbonate buffer. Okay, so what is happening is that the atmospheric CO2 tension, which will then dissolve into the cell culture medium to form dissolved CO2, which will form carbonic acid, which dissociates as H plus HCO3 minus. Okay. So the second component of this buffer is sodium bicarbonate, where we are forming the same bicarbonate ion. Okay, sodium plus HCO3 minus. Okay, so this basically forms what is called the bicarbonate buffer, and this is what usually helps maintain the pH of cell culture media. So it is very important that when we culture the cells, we maintain the 5% CO2 concentration because that is affecting the pH of our culture medium. Um, so as we discussed, our medium will have energy source. It has a pH buffer. It has amino acids, vitamins, and so on. But some cells require other components that we don't actually fully understand. We don't actually know what all is required for the growth of some cells. Okay, so often what we will add to cell culture medium is something called serum, which is the serum fraction of blood. It is usually bovine serum, so it's from cow blood. Usually we will add something called fetal bovine serum. Okay, so this is from fetuses. We also have calf serum and adult serum. And these will all differ in the nature of the growth factors and hormones, etc., that are present. So FPS contains over 1,000 different components, including growth and attachment factors, lipids, hormones, nutrients, electrolytes, carriers, enzymes, binding and transfer proteins. So FBS again does not have any defined composition. Okay, it will vary a little bit from batch to batch, but it provides lots of things which are necessary for cell growth, which we cannot provide in a defined manner. Okay? So normally, when you have medium, it will have like between 2 to 10% serum, because we cannot provide all of these factors in any other manner. Some cells, or rather, some media have now been created, some defined media which are serum free, where they have attempted to replace serum by adding all of these factors externally. So, if that is a good option for you, then you can consider going for serum free media as well. One thing that you may have to do if you're using serum is something called heat inactivation, which means you have to heat it to 56 degrees for at least 30 minutes. This is done mostly to inactivate the complement system. Okay, the complement system is part of our innate immune system, which is basically a set of proteins that can bind to foreign proteins and basically coagulate them. And in a lot of labs, this is part of the protocol for media preparation, that once you get serum, you have to heat and activate it and then use it. Um, it may or may not be beneficial, but often it is done. Okay? Normally, nowadays, a lot of the serum that you can buy on the market is heat and activated already, so you may not have to do this by yourself. Okay? So, that was the section on uh, mammalian cell culture media. So, now maybe we can discuss some questions related to that section. So, this question is, what happens to the culture of mammalian cell culture media, DMEM? DMEM is a very common medium. It's called Dulbeck's modified Eagles medium. This is a kind of basic mammalian cell culture medium. So it's asking what happens to the color of media when pH of the media decreases. So in DMEM, the pH indicator is phenol red. Like we discussed, when phenol red is present in an acidic medium, it changes to yellow. Okay. The pH of DMEM cell culture media is adjusted to blank. So again, DMEM is usually used to culture human cells, and human cells survive well at pH around 7, so it's usually adjusted to 6.9 to 7.1. The function of phenol red in cell medium is dash. So like we discussed, phenol red is present in cell culture medium as a pH indicator dye. Right? It is there to indicate visually if the medium has become more acidic or more alkaline. It is not going to act as an amino acid source or growth factor source or antibiotic or anything. It's just a pH indicator dye. Okay. While preparing mammalian cell culture media, student makes a mistake and ends up getting contamination. So which of these could be the mistake? Okay. So when we make the media, we have to dissolve the components in water and then we have to filter it. Okay. Generally, we do not autoclave mammalian cell culture media because since we're adding so many amino acids, vitamins, growth factors, etc., they will be destroyed in the autoclaving process. So normally we go for filtration instead of autoclaving. But when we filter the media, we should use the correct kind of filter. So 0.22 micron and 0.45 micron are referring to the pore size of the filter. Now 0.22 micron is a pore size which is smaller than any bacterial cell. So when we use a 0.22 micron filter, we are eliminating any bacterial contamination. But there are still bacteria that can pass through a 0.45 micron filter. So that is not going to sterilize your culture. Okay? So if we use 0.22 micron filter, we should not get contamination. But if we use 0.45 micron filter instead of 0.22, then there is a high chance that we will get contamination in our culture. So the mistake here is B, using 0.45 micron filter instead of 0.22 micron filter, because the pore size is so big that we ended up allowing contamination to pass through. Generally not autoclaving. Again, generally we do not autoclave mammalian cell culture media. We just filter it inside a biosafety cabinet. Okay. Why is CO2 often used in an incubator to culture mammalian cells? So CO2 is used often to control the pH. Okay. It is part of the bicarbonate buffer system, which is maintaining the pH of the buffer. Oh no, one of your dishes has bacterial contamination. What is the most likely reason? Uh, One reason could be that you forgot to add antibiotics. So normally in mammalian cell culture, people will add some antibiotics like penicillin and streptomycin. just to prevent bacterial contamination. But you should also practice proper technique and proper handling of the cells. So if you touch the dish, you should make sure that you're wearing fresh gloves, you're using ethanol, and so on. So you should practice proper aseptic technique in mammalian cell culture because it is very easy to get contamination in cell culture. The doubling time of bacteria is 30 minutes. The doubling time of a mammalian cells is 24 hours. Okay, so the bacteria will grow much more quickly than your mammalian cells, and they will completely overwhelm your culture. So it is necessary to practice proper handling when we work in mammalian cell culture. Okay, you're working with an epithelial cell line. Yesterday, when you split the cells, they looked good. So, just for reference, uh, an epithelial cell line, when it's adhered properly, it will start taking this kind of polygonal morphology. Okay, right? Because now it is stuck on the cell culture dish, right? So it's kind of flattening out and taking a polygonal morphology. So this is normal morphology. You're working with an epithelial cell line. Yesterday, when you split your cells, they look good. Today, they look like this. So here you can see all the cells are round. Okay, they're not adopting this kind of morphology, they're round. What could have gone wrong? So the cells are round. So that means they're not able to adhere to the culture dish. They are still stuck in the suspension. Now, what could be the possible reasons for that? Maybe they are not able to adhere because they've been incubated with trypsin for too much time. Okay? And trypsin has basically digested everything present on the cell wall. Sorry, everything present on the cell membrane, all the adhesion factors and everything. And that's why it is not now they are not able to attach properly. Okay? So most likely reason is they spent too long in trypsin. Trypsin has completely digested all the surface factors required for attachment, and therefore they are not able to attach to the support surface anymore. Yeah. So this was the segment basically on how to grow cells, how to grow prokaryotic cells, how to grow eukaryotic cells. Obviously, this is just a basic overview. You can grow all sorts of other cells. You can grow insect cells and so on. So the culture requirements will depend on the kind of cells you want to grow. But I think the basic principles still remain that you need to provide a carbon source, you need to provide a nitrogen source, you need to maintain osmolarity, you need to have a pH buffer, and you need to be aware of the nutritional requirements of your cell so that you can grow them properly. Okay. There is one more small topic in this section, which is differential centrifugation. So just give me 1 minute and then we'll get to that topic. Okay. So centrifugation is basically a technique that we can use to separate particles, and we can separate them based on differences in their size, shape, and density. So these three properties, size, shape, and density, together define something called a sedimentation coefficient. Okay, which is what we can use to separate particles based on centrifugation. So how centrifugation works is if I have some liquid and I have take say I took muddy water. I picked up some muddy water from a pond. When I pick it up, it'll look brown. right? Because there are all sorts of mud particles suspended in the water. But if I just keep it in a glass and let it stand for some time, what is going to happen is that all of these particles will settle at the bottom, right? Will settle at the bottom of the glass, right? This is where all the particles are, and this water will be clear. So this process is called sedimentation, right? And what centrifugation is doing is basically taking advantage of this process called sedimentation. So this process is going to take a good 20 minutes, 1 hour, a few hours, maybe depending on the size of the particles that are present here. So what centrifugation does is basically I can add this to a tube and then spin it at very high speed. So when I spin it at very high speed, okay, there's basically a centrifugal force which is acting on the particles here, which is going to force it to settle, okay, at the bottom of the tube. So it is basically speeding up the process of centrifugation, of sedimentation. Yeah? The tube containing a suspension of particles is rotated at high speed, which exerts a centrifugal force directed from the center of the rotor towards the bottom of the tube. This force acts on the suspended particles by pushing them towards the bottom of the tube, as we discussed. And the rate required, a rate at which they now settle at the bottom, is called sedimentation rate. So basically, how a centrifuge will work is that there will be some central point around which the tubes will spin. Here we'll have to add tubes into this rotor, okay? And a suspension will be kept in these tubes. And as the tube rotates, they will, the particle will experience a centrifugal force in this direction, okay? Tube is spinning in this direction, okay? Particle will experience centrifugal force, which will force it to move along the length of the tube. Okay? Radius, this concept of radius is important. That's basically the length of the tube, right, from the center of the rotor to the end of the rotor. This much is called the radius. So this centrifugal force is a byproduct of basically the particle being forced to move in the circular manner, right? As you can imagine, when particle is moving in a circular manner, it will have an instantaneous velocity which is the tangent to this point in the circle at this point in time. Which is basically the centrifugal force. Yeah. So the centrifugal force is basically the mass of the object into v square tangent, which is the tangential velocity, divided by r. As we discussed, that is the radius of the rotor, which is mass of the object into r omega square, where omega is something called the angular velocity. Okay, since we're moving in a circle, we need to consider the angular velocity in radians per second. So 1 rpm is 2 pi by 60 radians per second. Okay. So we can use this formula to determine the centrifugal force. So for for example, if we do this numerical, centrifugal rotor spun at 50,000 rpm. Okay, calculate the centrifugal force experienced by the particle at radius 5 centimeters. As a multiple of gravitational force mg, where g is 9.8 into 10⁻² centimeter per second. Okay, so basically what they're asking is what is the ratio of centrifugal force to gravitational force, where we know centrifugal force is given by m, which is the mass of the particle, r, the radius, omega square, whereas the gravitational force will be given by mg. Okay, so r we know is 5 centimeters into 50,000 rpm, which is 50,000 into 2 pi by 60, right? So basically converting rpm into this angular velocity, right? 1 rpm is 2 pi by 30 radians per second, okay, by g. And if we complete this calculation, we keep this in terms of g and we complete this calculation, then what we get is that F centrifugal is 1.3 into 10 raised to 5 into F grav. And what we would normally say this is that this centrifugal force is one lakh 39,800 into g, right? Or 1,40,000 g. This value is called the relative centrifugal force, or RCF. This is a very important number because the RCF value. Yeah, yeah.
Can you please explain again
Explain the numerical?
from the calculation? Yes, ma'am.
Yeah, sure. Sure. So basically, here what they're asking is they're given the speed of the rotation, okay, in RPM. This is rotations per minute. They've given the radius of the rotor, okay, and here they're asking what is this ratio basically of centrifugal force to gravitational force. So we know that the formula for centrifugal force is mr omega square. Okay? And we know the formula for gravitational force is mg, where m is the mass of the particle. So because mass is in the numerator and denominator, I've just cut it. Now the problem is in this centrifugal force formula, omega is something called angular velocity. Okay? Okay, but what they've given us, the information they've given us in the numerical is the RPM. So we have to first convert RPM to angular velocity. And so RPM is rotation per minute. So one rotation, right? One rotation just means I'm traveling around the circle once. So I'm covering an angle of 360 degrees, right? In one rotation, I'm going to cover 360 degrees. Angular velocity is in radians. So 360 degrees is the same as 2 pi radians. Okay, this is just another unit for measuring angles. So I'm just saying in one rotation, I'm covering 2 pi radians. So this is rotation per minute. And in 1 minute, we have 60 seconds. So one rotation per minute is the same as 2 pi radians in 60 seconds. Okay, so this is the unit of angular velocity. So to convert RPM into angular velocity, basically we have to multiply by this factor, 2 pi divided by 60. Okay? And so when we multiply, what now we're just substituting the value. Okay, so m is getting cut in numerator and denominator. R is 5 centimeter, we added here, and omega we are substituting from this value 50,000 rpm into 2 pi by 60. Okay, when we so all we have to do is this multiplication. When we do this multiplication, we get this value. Okay, 3.98 into 10 raised to 5 into f graph. Okay. So the other way to express this is just to say that it is 1.398 into 10 raised to 5 into g. Okay. It is 1,40,000 times the effect of gravity, and that is a value which is called relative centrifugal force, or RCF. And this is a very important value to know because RCF is the same value irrespective of which rotor you are using. Okay? It does not depend on the centrifuge that you use, it does not depend on the rotor that you use, it does not depend on the tube or a method that you've used. RCF is the pure value of the force that is being experienced. RPM can change. So if I have a rotor like this, a small rotor with radius 2 centimeter, and I have a bigger rotor, okay, with radius 10 centimeter. Okay, I can spin both of these at 2000 rpm. Okay, both of them are going to spin at 2000 rotations per minute. But if I keep my sample here and I keep my sample here, they will experience a different amount of centrifugal force. Okay, the amount of force here will be 5 times more than the amount of force here. Okay, because of this increased radius. But if I just say that you have to spin something at 2000 rpm and I don't specify the rotor, then there is no measure of how much force that I am applying on the sample. So actually the better way to express this is in this figure called RCF. So if I say spin it at 10,000 RCF, then I can set both of these rotors to 10,000 RCF. Okay, the actual RPM value will be different, but the amount of force that my sample will experience will be the same. Okay, so you may see in protocols, someone will tell you centrifuge at 10,000 G. Okay, this is the RCF value, which is independent of the rotor and the centrifuge which is used. So we may be familiar with just like normal centrifugation, but there are many kinds of centrifugation that we can use. So one is something called sedimentation equilibrium centrifugation, okay, where we try to estimate the molecular weight of the substance. So how that does is we are just trying to measure how long it takes for the molecule to sediment, okay, which is related to its size and so on. So we can try to estimate what is the molecular weight of a compound through centrifugation. Then if you want to, you can imagine in sedimentation, everything is going to be collected at the bottom of the tube. But what if I want to make different fractions of my sample. Okay, I know I have a very heterogeneous sample. Okay, there are things of many different sizes and many different densities. If I spin this at a very high RPM, then all of these will come here in my pellet fraction. But it is actually possible to separate all of these by spinning them at different speeds. So, one option is that I can spin them at different speeds. So, first I'll spin it at a low speed so that this will come out. Okay, first these big particles will sediment. Then I can spin it at slightly higher speed and then at an even higher speed. Okay, and that way I can sort of separate the different components of the substance. The other option is something called gradient centrifugation. Okay, where I can have a tube and here I can have some liquid in the tube which already has a density gradient. And because the substance was moving in a density gradient, it will get stuck. Okay, different components will get stuck at different levels which are equal to their own density. Okay, so we'll discuss this equilibrium density gradient centrifugation, okay, in some detail. So um, basically ultracentrifuge is a centrifuge that will spin at very high values. So this will be something which is spinning at say 50,000 G, 100,000 G. Okay, and that means when you're applying that much force on your sample, that means you can sediment even very small subcellular components. In your normal centrifuge, you might have a centrifuge in your lab which will go up to maybe 15,000 RPM. And you will use that for sedimenting, for pelleting cells. But if your components are much smaller than cells, like some subcellular organelle, to force that to sediment, okay, you need to actually spin it at very high speed so that a lot of force is applied, a lot of centrifugal force is applied, and it will come out of the solution. And for that, you need something called an ultracentrifuge. Ultracentrifuge can be of two types. This is something called fixed angle. Here you can see that there are these slots for the tube, and it is spun when the tube is held within the rotor like this. Okay, and because the angle of the tube is fixed, you will get a sedimented material here like this at an angle. Okay, so when I take the tube out like this, the sedimented material will be at an angle on the side of the wall. The second option is something called swinging bucket. Okay, where basically there's a hinge over here and the tube is free to move and spin like this at a 90-degree angle. And what you can see is that because of this, the radius of the rotor has increased, right? Here the radius of the rotor is this much. But because this has been allowed to come all the way out over here, the radius of the rotor has increased. So the amount of force that is going to be applied, amount of centrifugal force that is going to be applied is more. This swinging bucket rotor. Okay. Also, the centrifugal force is applied directly like this. So when we remove our tube from a swinging bucket rotor, the pellet will be formed exactly at the bottom. Okay, it won't be on the side, it will be exactly at the bottom of the tube. Okay. So in most preparative ultracentrifuges, you will see something like this. You will see swinging bucket rotor. Now, one application of centrifugation or differential centrifugation is something called self-fractionation, which is when we are going to collect different fractions of cell organelles by centrifugation. So we can start with some tissue, okay, which is made up of different kinds of cells. Okay, say I took some liver tissue. from the liver tissue, I want to study what the mitochondria and this liver tissue are doing. If I want to isolate the mitochondria from this tissue, then I mean, you can think that it doesn't sound like the easiest task. No, it's not like I can individually go and pick up the mitochondria in each cell. So instead, what we can do is first take the tissue, and then we have to break up the tissue. Right? We need to get rid of this and release the individual cells. Okay, so that can be done through things like enzymatic treatment, homogenization, and so on. And after we've released the individual cells, then we need to break the cell. Because you want to get subcellular components, you have to lyse the cell. So you can lyse the cells through sonication, and then what we're left with in the tube is different cell components. Right? We will have little like broken cell membranes, nucleus, mitochondria, ribosomes. All of these are just going to be floating around in the mixture now. Okay? And you can think that all of these are of very different sizes. Right? So you can think that the nucleus will be quite big compared to a mitochondria. And the mitochondria will be quite big compared to a ribosome. Right? So we can take advantage of these differences and separate them. So here in the cell homogenate, right, which is what we have in this step. In cell homogenate, first we will do a low speed centrifugation, which is 100 G. 1000 G, 10 minutes. Okay. And here in this pellet, we're going to get whole cell, nuclei, and cytoskeleton. So all of the large components of the mixture are going to come out here. So these are unlysed cells, which are going to be the biggest component. The nuclei, again, which is very big, and any cytoskeletal material. Because these are big, we don't need to apply a lot of centrifugal force, so we can do a low speed centrifugation to remove these. So now all of this will be confined to the pellet. So if we pick up this supernatant portion, this will still contain a mixture of all the other subcellular components. So we can transfer this supernatant to a fresh tube and centrifuge it again. So here we can say supernatant subjected to medium speed centrifugation. Okay, this is 20,000 G. Right? Now we've increased the speed. And because of this increased speed, we're going to get a pellet of the next set of organelles, which are mitochondria, lysosomes, and peroxisomes. So these are the membrane-bound organelles, right, which are smaller than nuclei, okay, but they're bigger than things like ribosomes. So this is going to be the mitochondrial pellet. So if we were interested in mitochondria, we would stop over here. But maybe I want to study ribosomes. Okay, I want to study the structure of a ribosome. So I have to isolate that specifically from this mixture. And for that, again, I can take the supernatant part and subject it to in even higher speed centrifugation. Okay, this is 80,000 G. Okay. After this, I will get small vesicles. And now to pellet everything else which is left in this mixture, we have to go for extremely high speed centrifugation. This is 150,000 G. Okay. For 3 hours. Okay. So this is an enormous amount of centrifugal force being applied, and you are forcing the small particles to precipitate or sediment at this point. Okay, and then you're going to get a pellet which contains ribosomes, viruses, and large macromolecules. Okay, and so in this way, we've able to separate the cell into a number of fractions, okay, based on their size. So here you can see typical centrifugation parameters for biological sample with the radius of about 5 centimeters. So for analyzed cells and for nuclei, we can you work at relatively small values of G. Okay, these are things we can do in uh, benchtop centrifuge. Okay, but if we want to pellet smaller and smaller components, we have to go for vacuum ultracentrifuges. Okay, so these are things that are going at 1 lakh G, right, for a long time. And ultracentrifuge is going to require a vacuum and it's going to require cooling, okay, to make sure that the system does not overheat, okay, and that the system is able to run at such high speed for such a long time. Okay. So these kinds of separations are only possible in an instrument called an ultracentrifuge. So after spinning at 1 lakh G for 2 hours, you will be left basically with the cytosol in the supernatant. And your pellet will contain the ribosome, small polyribosomes, and some soluble enzyme complexes. Okay. So what we discussed was basically sequential pelleting, right, of the substance. Basically, we were doing many different spins, and um, at each step, we were getting a pellet and a supernatant, and we were going to discard the pellet and collect the supernatant. But if you want to do this, you can see this whole procedure is going to take a very long time, right? Because it's going to become very tedious to remove these things one by one. So, what if we can separate them all within a single spin? So, for that, you need to do something like gradient centrifugation, okay? Which means in this tube, in my sample, I'm going to add a gradient. So, this is going to basically allow different densities to form throughout the tube. I'm going to add my sample on top. Okay. After centrifugation, what's going to happen is that as it is impeded by, so essentially, as it moves through the more and more dense layers, it is essentially going to be stopped at the stage where the density is equal to the density of the organelle. Okay. So if I have some less dense organelles, they will come over here. If the density is more, they will come over here. And then basically, what is going to happen is I'm going to separate the mixture into different layers. And then what we can do is just poke a hole in the bottom of the tube and collect this liquid as fractions. And so we can see that we're able to separate these pink and black spots because they have different densities. And so in this process, we can collect them as different fractions. Okay. So obviously, depending on the nature of your sample, you have to choose your gradient. So this is a shallow gradient, which is 5 to 20% sucrose. But if your sample has very different, uh, there's components which are very different to each other, you may have to choose something like a steep sucrose gradient. For example, 20 to 70% sucrose. So this is going to give you a bigger range of densities. And so you can separate more effectively. Okay. So look something like this. You'll make your gradient You'll add a sample on top, centrifuge, okay, to ensure that for 8 to 15 hours, to ensure fractions have reached their isopycnic points. Isopycnic is equal density. Okay. Reach the point where density is equal. So here you can see this sample, which is cell homogenate, has been separated into a plasma membrane pellet, and then we can have different membrane fractions, okay, and mitochondrial membrane, endoplasmic membrane, and so on. So here you can see we are able to separate this mixture into different fractions depending on the density. If I have an unknown sample and I want to determine what is the isopycnic point, then I can also do that through this experiment where basically you have to collect the fractions and it collect the fraction, then you can determine the sucrose concentration in each fraction, which will tell you the isopycnic point of your organelle. Right? So, for example, if we have here, they've used a collection of different proteins, okay, and what they've done is collect different fractions, okay. You can see amount of sucrose is increasing, okay. So this is the first fraction. The fraction is at the bottom of the tube, and these are the upper fractions, right? So tube looks something like this. So 1, 2, 3, 4, 5, 6. Okay, and here you can see in the first fraction we have not gotten any protein. In the second fraction we have gotten mostly this pH 6.5 ATPase. IDPase has come mostly in the mostly come in the fourth fraction, and this NADH cytochrome C reductase has come mostly in the fifth fraction. Okay. So this here they've used it to separate three different proteins. Use a sucrose gradient from 20% to 45%, and that has allowed us to separate these proteins just on the basis of their density. Okay. Now, if you want to improve the resolution, you can use something called a discontinuous gradient, where basically you're going to make these layers. Okay, and because it's not a continuous gradient, it is not going to settle at the exact point where the density is equal. Instead, it is going to settle above the layer which has greater density. Okay, say the isopycnic point of my sample was at 32% sucrose, but there's not going to be any 32% sucrose point in this gradient. Instead, my sample is just going to form a layer over here above the 34% sucrose layer. Okay. So we see fractionation at interfaces between sucrose layers based on density of organelles. Okay. So this is a good way to improve the resolution of the layers. If you already have an idea of what kind of gradient is required for the separation, then we can make a discontinuous gradient. How to recover the sample in this case? I already know that this is the sample that we want. So use a needle to puncture tube 3 to 5 mm above your desired organelle. So puncture here and then just withdraw the purified subcellular organelle fraction. Okay, so we can just isolate the fraction that we want. We already know we don't need these two, we only want this. So we can isolate this with a needle. So yeah, so this is how it would work. Here we have increasing density of sucrose. So this is done by making various concentrations of sucrose. So this is 1.09 to 1.25 grams per centimeter cube. Why are we using sucrose? Because sucrose gives us a good range of densities which is comparable to densities of our organelles. Okay. So now when we add the organelle fraction and then have centrifugation, you can now see that lysosomes, mitochondria, and peroxisomes, if you remember in our first pellet, okay, all three of these were separating together. Okay. After this medium speed centrifugation step, you're getting all three of these together in the same pellet. But if I want to separate these, I have to use density centrifugation, because I can then make use of this subtle difference in the density. So lysosome is 1.12, mitochondria is 1.18, and peroxisome is 1.23. Okay, and they will then settle at these interfaces at the appropriate density. Okay, and then you can just use a syringe to isolate this mitochondrial fraction. So here you can see um, size and sedimentation properties of subcellular components. You can see that different subcellular components require different kinds of RCF. So combination of the centrifugal force and time is what is going to force these to pellet. Okay. This I think we'll skip, but we should just be aware that there is something called the Swedberg equation. Okay, and what the Swedberg equation does is it allows you to predict the molecular weight from the sedimentation time. Okay, this is something called the sedimentation coefficient. Okay, and what the Swedberg equation allows you to do is predict the molecular weight of of an unknown molecule from its sedimentation time. Okay. Yeah. So here you can see protein has sedimentation coefficient of 7.16 s. From that formula, we can calculate it has molecular weight of about 14.5 kilodaltons. Okay. And this S is the same S that you see in like 70S ribosomes. Okay, this is the same S. Okay, it's named because of its sedimentation time. So just in the last section, we'll go through some questions for this. So in a certain study performed by a group of students, unknown cell type, they've done centrifugation, they yielded fractions A, B, C, D after these 4 steps of centrifugation. So step 1 is 600 G, after which they've received fraction A. Then 10,000 G, 100,000 G, and 32,000 G. So 6,000 after 600 G, so that should be the heaviest component. So after this, they should have got the nucleus. Right? After this, they should have gotten a slightly lighter component like mitochondria. After this, they might get things like ribosomes and plasma membrane. So component A has to be nucleus. Okay. Component B will be mitochondria, C will be ribosomes, D will be plasma membrane. The centrifugal force and time required to carry out separation of mitochondria from whole cell lysate is dash. So mitochondria requires a sort of medium speed centrifugation, which is something like 15,000 G for 15 minutes. It will also pellet at this kind of centrifugation, but it will pellet along with a lot of other things. So with this you will get ribosomes and other things like that. But in the fractionation pathway, kind of mitochondria will come at this point. Okay, this is for nuclei. The most appropriate use of detergents in the process of differential centrifugation is dash. So detergents are used to lyse the plasma membrane and extract the cell organelles. Okay, detergents will basically break apart the lipid plasma membrane and extract the cell organelles. The medium used in cell fractionation should not be dash. It should not be hypotonic. Okay, we should not do not want to lyse your organelles in the process of cell fractionation. Treatment to remove the cell wall from plant is cellulase. Plants have a cellulose cell wall, so you can remove it with cellulase. Okay. In the isolation of the periplasmic fraction of the prokaryotic cells, which substance is used to give osmotic shock to the cells? So, in this, basically, this is part of a question for a protocol. So, the substance or action used to give osmotic shock to cells is first we have to incubate it in a hypotonic buffer and then followed by centrifugation. Okay, and just one last question. Um, Tropomyosin, okay, this is this long protein. This is 93 kilodalton. Hemoglobin, which is 65 kilodalton. Okay, you can see it's a much smaller protein. What you would expect is if a protein is bigger, it should sediment faster, right? It's heavier, kind of. But they are saying that hemoglobin actually sediments faster than tropomyosin. Okay? These How is it that the bigger protein sediments more slowly than the smaller one? Okay, and the trick to this question is you have to think about what is the shape of these two proteins. This is a very elongated protein. This is a very small compact protein. And what because of its this is large surface area, it is actually going to face more friction. Okay, there's more surface area exposed to the medium, and it is going to face more friction as it moves through the medium, which is going to oppose its sedimentation. Okay, so which is why this is going to actually sediment slower than a globular protein like hemoglobin, even though it is actually a heavier protein. Okay, so that is as much as I wanted to discuss today based on the week 8 syllabus. Um, if you have any further questions or you want to discuss anything else, I am open. Or otherwise, of course, you can find the recording of this on YouTube if you want to ever come back to it at any other point. If you have questions in the middle of the week, feel free to use the Google group, which is there in the Q&A tab in the course. But that is all from my side tonight. So, yeah, good night.